Thank you so much, and sorry about the technical issues. Um, yeah, it's a pleasure to be here again for the second presentation of the day for me. Today I will talk a bit about the wireless EEG recordings in sports and what kind of applications people usually use by inducing EEG recordings into a sports activity. Um, the main question would be, why would you actually want to do that, right? Why would you want to see EEG traces on someone practicing sports? Actually, there's a lot of reasons. Obviously, the first one is to actual measure physiological changes in the bodies during the physical activity. So you can this could be very, very informative, for example, on athletes that are going to do extreme sports. For example, like you see here, Christoph hiking into a very snowy mountain and staying there for a long time. So you can see him using a wireless device, a G.tec, with the dry electrodes. This can be very useful for the measuring the physical activity, how our body changes and adapts to the physical activity in sports, and can be a very good indicative not only in the assessment and the diagnostics part, but also in a general way to see how our brains develop and adapt to such circumstances. You can also use EEG to compare professional athletes to amateurs. This is widely used. We see a lot of papers using that where they take a group of professional athletes, measure the EEG during the performance of the sports, and then take amateur athletes, so people that don't do it professionally, or even people that don't do it that often, to and compare how the brain activity acts in between these two groups of people. Very, very interesting kind of measurements. Then we also have one of the biggest amount of studies, which is predicting the performance of athletes. Um, this is historically done because of the limitations of EEG, and we were going to go a bit further into detail in the next slides. So one of the biggest ways to predict the performance of the athletes is actually measuring their brain activity, their physiological measurements before the performance of the sports, most of the times one day before or during the night before the event, and take conclusions. Most of it because of how well rested the subject is, if nervosity plays a big role in this particular athlete, or for example, if this person is able to have a very restorative night before the sports activity. We also have then more kind of a BCI approach to EEG with sports, which is related to training and improving performance of athletes. We see this a lot with several biofeedback kind of applications where you have an athlete, the athlete wants to outperform all the other competitors, and how can we do that? Uh, how can we perform several activities or tasks in order for the athlete to be able to perform better on the next day and outperform all the other athletes? So you can take EEG traces, try to improve them, try to make the subject able to concentrate more, apply different kinds of applications so that you can see these EEG traces and how the person is developing across the time. So this is one of the biggest functions where you will see EEG applied to sports research. And then obviously the last one would be finding new biomarkers to be used in either clinical setups, assessment, diagnosis, but also that will be used in future for future applications of EEG sports research. This is something that is widely important, the finding of new biomarkers, because it will allow the researchers to have a better overview of the performance, cognitive performance, and also the end sports performance of the subject. So what kind of EEG markers do people currently use? So you have the typical ERP, so the auditory evoked potentials, visually evoked potentials. You basically want to induce these potentials on the athlete in order to be able to see how big these peaks are and how good they perform while performing sports. So, for example, you want to distract them, or you want to tell them to be concentrated on a particular stimuli. It can be an auditory evoked potential while they're doing sports, and if they can do both at the same time, is a very good indicator that they will perform well because their concentration is just there. You have the motor imagery, which is seen a lot in studies, for example, for table tennis or golf, where instead of having people actually performing the task, you are showing them images. For example, a tennis player, you show a video of the other athlete throwing the serve, and then he has to imagine how we will perform the motor imagination of the strike back to the other athlete. This is something that we see a lot in studies. We see spectral changes. By this, I mean changes in the beta gamma activity during the performance of sports, on the alpha activity, on the performance of resting state EEG. We have, like I mentioned a bit before, the sleep studies, which will tell how restorative the night before the big event will be, and this can have very big influences on the performance of the athlete. And the combination, obviously, of other biosignals, for example, respiration, galvanic skin response, EMG, EOG, eye tracking, so you can find out if the person is having a very good respiration rate. You can see how nervous or how calm the person are with a galvanic skin response. See reaction using EMG, for example, the time that it takes the subjects to perform an activity, and we can see with EMG how fast the reaction time is, and the eye tracking and EOG where actually the person is looking like. So you have, for example, a goalkeeper in a football match, and you can measure where they are looking at, if they're looking at how the foot is of the athlete is stepping on the grass, where the ball is coming from, and really detect how good they are at scanning, for example, during the match. Which are the typical problems of the studies? And this is something that is mostly related to the technical aspects of this EEG research. Mainly, the biggest issue is the movement artifacts. Movement artifacts can really ruin any kind of measurement. You will have it ruining the quality of the EEG, and this can be extremely problematic. So it can complicate to make it totally impossible to have any results. So one of the things that in the past people have done is actually choose either sports with low movement. So for example, golf or chess, where you have a stabilization, especially on the head. No, you have a stabilization, so the head will not move a lot, and this will not induce a lot of movement artifacts. Or people choose to simulate the activity. So they will show videos, they will show images, they will show virtual reality setups, meaning that the people will not be running around. People will be staying still and will pretend that they are playing. And by doing this, you'll be able to measure some of the typical EEG activity that you would potentially find out on an EEG measurement during the sports activity. Obviously, this is not perfect, and this is cost because most of the EEG devices, especially in the past, had cables. And cables with the movement, you were able to detect a lot of artifacts. And also the people were moving around with heavy devices. So we see a lot of papers being released with people either wearing a big backpack with a big device on their back. You have cables hanging around everywhere. Or in case, for example, of this photo that you're looking at, if you don't have a wireless device, you have cables going up to the ceiling, and then after going to the ceiling, they will go with several meters up to the amplifier and the computer, which obviously is not ideal because not only, especially on cases where you don't have active electrodes. So if you don't have an active electrode or a preamplifier very close to the source, the cables will act almost like an antenna and capture a lot of noise from the surrounding. A lot of movement artifacts from the moving, from running, will be detected as well. And this is almost impossible to have a very good EEG quality signal. Then one of the other problems is obviously the lack of concrete biomarkers. Okay, you have an EEG, it can be perfect, but what do you want to look at? What kind of traces do you look at to be able to measure performance, to be able to measure attention? How can differences in different athletes represent itself on the EEG? You're looking at how active, or for example, amateur and professional athletes compare. These brands will be very, very different. How can you actually differ between these two? And this is lack of these concrete biomarkers is obviously one of the biggest issue, and it is something very widely discussed, especially in the beginning of the papers. What kind of biomarkers are they going to use? If they're looking at the spectrums of frequencies, are they looking at evoked potentials? Are they simulating the user? This is something that still is very limited, but as I said, with the creation of new biomarkers, this definitely will also improve the quality of the papers of EEG in sports. And a combination of all, obviously, during decades, technology just did not allow to do this in field studies. So we don't have EEG devices that are wireless since a long time. So this limits the amount of papers that are available, the amount of literature that is available. We have a big boom in the last 10 years because these kinds of devices started to appear and people start to apply it in these kinds of studies. So this is where the wireless changes the paradigm of the study. So we can see here one of the first tryouts that we had. So we can see Martin, who was presenting in the morning, just going on a treadmill, walking, and we are trying to detect if a wireless device, in this case is a Nautilus, can be able to uh, produce enough or good quality EEG enough for the system to detect P300 patterns on his brain, on his visual cortex. So he's doing the calibration right now. You can see that so we have these items here on the top. Let me you have these items in the top saying the quality of the signal. If there is a lot of artifacts, and you can see as he walks, there is a bit of artifacts, but still we can see the selection. So we will try to look at here and we will see how quick the selection of each individual item is being performed. So he is able to select this one, he's able to select this one. Now he tries to select this one. Nice. Tries to select this one, etc., etc. So you can see already a big change on just having a wireless device can already be a big plus on these kinds of recordings. And then obviously there is another big thing that we have been seeing coming into the markets in the past years, which is the artifact removal tools. We see here, for example, Martin on the spring school 2 years ago. Walking, jumping. On the left side, you see raw EEG, so without any kind of filter, without any kind of, no, sorry, it has filters, but not cleaned with the Oscar. And on the right side, you see the Oscar correction. So you see how EEG is impacted by the brusque movements of the body. So he's doing some squats. And you can see already just the squat, even though the head is a bit stable, already provokes some shifts on the EEG, but Oscar is able to clean, which is very, very good. So you can see how these kinds of applications can be very, very useful. So he's doing some push-ups, which provokes vertical and lateral artifacts. He's going to do a handstand. And then you can see how the EEG is completely destroyed without this artifact removal tool. And on the right side, you see a very clean EEG. Obviously, this artifact removal tools have to be very, very neat, because uh, you can you can remove artifacts, right? You can remove all the movement artifacts as you want, but if the artifact removal tool also removes EEG traces which you want to investigate, then you will have a big issue, because you will not be able to do any kind of EEG measurements if they're gone. So this is why in the morning, and I also stopped to see that you can see the backflip where the EEG is completely gone, but Oscar could correct it. Like I was saying, if you don't have these traces, and the EEG can look very good, but if you don't find the actual EEG traces of the brain of the athlete, then it's useless. So it's a combination of being able to read the EEG traces and being able to have very good quality of EEG. So these were some of the past examples, because we are going soon to the practical session. These are a couple of past examples that we've done in this session in the past years. So in the first time we were started doing some EEG measurements for sports using a static bike. Static bike, obviously, the same issue as usual. Solved. You don't have much movement, so you have someone just standing on a bike doing some sports. We used at that time a GUSPM, which was possible to place next to the user, because the bike is static, will not go anywhere. And we were sending through audio with several boxes, audio triggers delivered via Simulink to the subject. And we were also trying to also induce other stimulation, like visual stimulation. Why audio stimulation? This is something that we're going to also use today. They serve as distractors, not only that, and so you can see also how concentrated the subjects are, and also are a very good indicative of how good the EEG quality is, because if the EEG is bad, you will not see any auditory evoked potentials. Then we have Catherine, who will be also doing the session today. In 2022, we decided to do something a bit more different, a bit more free. We were using a g.Nautilus, which allowed Catherine to be able to move around. We did some martial arts, because she's a big fan of martial arts. We did some auditory evoked potentials through Simulink, through a tablet, through a box, until Catherine through headphones. So you can see the setup how it was. It was a running backpack. We had a tablet, Microsoft tablet. We had a hub where you can connect the G Nautilus base station. We had an auditory trigger box sending the triggers directly to through a cable to the receiver, and sending the audio information to the subject. So you have the box here sending the information or the auditory evoked potentials to the ears through the headphones, and sending it with the cable to the base station. Everything is mounted then on the backpack. I decided to put the tablet outside so that I can start and stop the model. I can check the EEG quality. So this is how it looked like. I used some cable binders to hold it into the running backpack. So this was the setup: an Nautilus, an auditory trigger box, a trigger cable, headphones, and then there was no cables between the amplifier. Was neatly fitting inside the backpack, and the amplifier sitting on the back of the head. And this was a simple model that we were using, with the auditory stimulation being sent through Simulink, and you can do a visualization in real time. And this is a video of how the first session, which people loved it, and therefore we're doing it every year, of Catherine fighting, and still we can see how the EEG activity is looking like. Nice. So in 2023, we decided to do something a bit different, not on evoked potentials, but on the concentration level during the physical activity. Because many of the studies, like I mentioned, do that, and you can also see a lot of papers mentioning how alpha activity is increased if someone is, for example, attentive and aware. So like you see in this paper here from Sharon et al., relaxed but well-focused athlete would demonstrate large alpha amplitude before accomplishment of the motor tasks. So we try to apply this concept with our colleagues. So they were two people playing badminton. There's a lot of movements compared to most of the sports, and there was concentration necessary. So we were using a g.Nautilus. We were not doing any evoked potentials. We were just performing a classification task before. So they were trying to do cognitive tasks, like clicking on the button and trying to memorize it. And then we did a resting state. This would classify this particular user. And then we were looking at the alpha modulation in real time. So we can predict if the person is able to concentrate and able to perform well. Then in 2023, we used the raw data and filtered it with the frequency bands, and we gave the feedback with the concentration bar. So as I said, they were playing badminton. They pressed this button, cognitive tasks. We looked at the EEG, and then once we said start the game, they started playing, and then there was a biofeedback bar going up and down depending if they were concentrated or not. So we had two bars and we could see if they're concentrated or not. Then last year, what we did was there was Oscar already on Simulink, so we decided to try to have two PCs. Martin was using was using a unicorn. He was doing several sports activities like you saw in the past, and we were sending via UDP from the subject's PC to a more complex PC where you have a camera, you have your screen sharing, you're processing all the data. And the two computers were communicating with each other, and we were able to see how the evoked potentials were looking really neat. So this was the model that I used last year. So you have a unicorn sending the data, very, very simple, and then you could see evoked potentials in real time. So you can see here some of the evoked potentials that Martin had, so very big peaks of P300 while he was performing some tasks on the floor of our seminar room. And what will we do this year? So now we go back to the session, the demonstration session. So this year we decided to go with the biggest or the most used sport for these kinds of studies, which is table tennis. Table tennis is neat because you have a lot of movement. But you are still able to perform different cognitive tasks. It requires concentration, but at the same time, it also requires movement from the side of the athletes. I have two of my colleagues here, and we will be starting the presentation now. So I will just stop sharing so that I can show you the screen. I will use the other camera. Let me just jump to the other camera. So we can already see Evgeny. And we have here table tennis, the table of table tennis. We have the two amplifiers. I will ask maybe for you guys to come to this side so that I can set up the let me exactly. So one by one, we are using the g.Nautilus Pro. We'll use two of them. And you will use my computer that is streaming and receiving the data through the Simulink block. So one for Katrin, one for Evgeny. Let me place on the back. Actually, will need a holder for you. Let me get the syringe. Oops. This one, yeah, thank you. And I'll start gelling you, and then Evgeny will be the next one. Let me start gelling. So I'm using active Whoa, sorry about that. Active electrodes, G scarabeo. I will just place it. So it's very important to have the correct setup because you want it to be very neatly. Exactly. So I'm gelling all the electrodes using a 16 channel amplifiers. And we'll be able to see in real time. So we'll have one computer receiving the data from both amplifiers. You can use this on Simulink. On Simulink, you have the block for the amplifiers, and this block will allow you to capture more than one amplifier. I will use also on Wednesday in the morning for the hyperscanning. If you're interested, please go to the hyperscanning session because it will be a very interesting session where we will use the maximum amount of electrodes, of amplifiers, sorry, with several people, and we will introduce a surprise, which is animal EEG. So we'll have Santi, the company stock, Christoph stock, to induce some excitement into the session and also be able to see if we can see the biggest difference between how auditory evoked potentials on a dog can differ from a human auditory evoked potential. I think I have them all. I will just go just a second here. Um, Catherine is applying also gel so that I can be a bit more concentrated in talking. So we will do a little. So I'm applying also the amplifier to Catherine and connecting it. Very good. Catherine is applying gel to Evgeny. So what will we do today then is I will go to the model, but the main big thing is we have the two rackets, so they will play with each other. I'll drop Catherine's already on the table. And then we will be able to see if they are concentrated during the whole session. I will ask them in the beginning to play really slow so that we can see how that affects the signal and the quality of the evoked potentials, and then ask them to potentially get faster and faster. And I will try to make their lives even more complicated afterwards. Let's see how they perform. So we're almost finished. So you can see pretty quickly, you can set up 2 people. If you had 2 people doing exactly the same thing, would be even better. Yeah, thank you, Catherine. So on this new version of the caps, you can see there is a magnet. You just put the magnet and connect the amplifier. And I think you're ready to go. Thank you, Evgeny. Now it's my time to sit because I'm getting old. And we have here Catherine and the rackets. In the meantime, I will share again the screen because I want to show you the model that I am using, just to explain you what is going on the side of the computer's processing. So this is the Simulink block. I hope everybody can already see. We have, as I said, the block of Simulink, who is having two devices. This is already starting to be a mess. It's feel like We have two. You can see each amplifier is identified by the serial number. Uh, in this case, I wanted to see evoked potentials for the two different people. So we have the evoked potentials for user one and the evoked potential for user two. We are calculating it, throwing everything into the Oscar Live block, which will then take the data, try to remove all the movement artifacts, and then send the cleaned data to two scopes, one per person. And after that, we can have two files. You can decide to have one as you wish. In this case, I'm recording two different files because I want to study the person individually. And after that, we are going to see the evoked potentials. We have here the most important part, which is the paradigm presenter block. The paradigm that we're going to use is an auditory evoked potential paradigm, which takes around 8 minutes long. Is giving a non-target, which is a low tone beep, and a target, which is a high tone beep. The high tone beep will appear randomly, and then we'll be able to see how they are performing. Okay, let's go. I can already stop sharing so that you can see a bit more of the action that is going on. You guys can start. Oh, there is that's the 10th. It's okay. Just let it. Yeah. Okay. So you can start playing while we are loading Simulink. Let's see how they are performing. Let's see which one of the two is the best. So yeah. In the meantime, we're loading the paradigms. Let me have a look. So what we're going to see is I am closing in. Good. Let me have a look because I will share now the screen. We will have system running. Let me just see. Okay. Let's open the devices so that we can apply the devices here. So you can see the two devices are being recognized here. You just press Control and then you select both of them. One of the most powerful thing about this G Nautilus Pro is that even though you have an amplifier with 32, you can always use 16 channels if you have a 16 channel cap. So what we will do is actually look at the configuration file, and then we'll be able to load it a 16 channel montage. My Zoom is a bit killing making it very slow, but we will have a look here. For the evoked potentials, what we are currently using is the paradigm presenter. So you can see this black box will show the presentation. You have seen this also on the ECOG presentation of CAPI, but also on both the presentations of Christoph and of Martin, where you're able to see how the presentations will look like here. You have two screens, one monitor for the subject and one monitor for the user. And it's very easy. The paradigm presenter, you can have an XML file with your presentations. You can load videos. You can load images. etc. So it's very, very neat, and it's very useful to do the to do all the tasks in one time. Let me just, as usual, presentation syndrome. Things are not working as planned. I am just restarting the g.Nautilus so that we can connect better. Let me have a look. One Nautilus is connected. The second one is also connected. Let's try to connect them again. And then we connect 1 and 2, select them. And once we have them here, we will be able to run it. So I can show you also a bit of the paradigms in the meantime. So with the paradigm, you can do different kinds of applications. So if you have the g.tec, the paradigm presenter will be showing up on g.tec, g.tec examples paradigm presenter, and then you have already preset auditory or eye tracker or finger tapping, so paradigms. And then you can see here the different tasks and different paradigms that we are currently working at. Let me just close the model and open it again. And MATLAB, hence. So we can try to open it again. Zoom is usually making things complicated, but it will work now. So let's connect it again. And once the configuration will pop up, we will be able to select both. Yes, okay, now it worked. So MATLAB plays some tricks. So we have 2 users, 16 channels each. I am applying a bandpass filter and a notch filter, 0.5 to 30 Hertz and 52 on both. So you can see on the top both of the amplifiers, and then I will press OK. We are now going to the paradigm presenter. You have the imagination, the monitor here. We can select the different paradigms by selecting it on the bottom line here. So I will select ADP UDP Sports, which I created for this purpose. And that should be basically it. Let's run the model. And we'll be able to start seeing and hearing the auditory evoked potentials. In the meantime, they're having a lot of fun. Well, okay, we can see it's running everything. There is the fixation cross. Maybe you can already start hearing. We will look at the different EEG. So this is from Katrin. And let me open the second one so that we can compare. So very nice. Let me remove this window. So you can see they're moving a lot. One of the Oscars is working pretty neatly. So I can see already the evoked potentials popping up. Let's see if there is any interference with that. I will open for both people. I think I opened for both. One of the great things that you can have by having separate is that you can also apply a montage. So you can see the different electrodes, electrode number 10, 9, 12 in the positions that they are placed. And then you can go inside and have a look at the evoked potentials in real time. So we have an overview of both subjects, clean data of both. So let's have a look here. Let's make a very neat. Let me also now change the stop sharing so that you can see a bit what they are doing. Let's see if you can have a look at the different. So you can see they can move around pretty easily. They don't have, so all the devices are currently connected to my PC. So there is no PC close to them. I mean, I'm a couple of meters away, but you can see clearly that the device does not provoke any kind of difficulties in terms of the movement of both subjects. So you can see the auditory evoked potentials. You can hear the beeps. It's typically very hard to concentrate using the beeps and looking at the screen. I'll share the screen again. So that you can have a look. So even though there is a lot of movement, you can see that the EEG is being cleaned very nicely. Guys, can you stop just a second? So you can see the EEG artifacts are pretty lower, so you see much neater how the connection is like. Can you close your eyes and relax? See some alpha activity. Can you open your eyes? Okay. So let's have a look again. So you can see both signal looking neat. And let's start playing again. You can see some of the movements, but still the EEG looks pretty fine. Keep it. So yeah, you can see that you can do a lot of complex stuff by using this model. So you have the amplifier, you have the Oscar, which is trying to remove all the different artifacts. You have two different kinds of visualization in real time of all the EEG. You have visualization of the evoked potentials with also an evoked potential calculator block, which is taking the group ID coming from the paradigm presenter directly into the feed of the evoked potential block. At the end, you can take then the two separate files, and then of course you can analyze it more thoroughly offline. This will be widely important in case you need some kind of adjustments or you want to have an overview of the device. Okay, there seems like they are pros. And I guess we are about to finish. So I would like to thank my two subjects. You guys can play, but thank you so much. And I guess we will stop also the model so that we can stop hearing the beeps. I will change the camera and I will thank you so much for the opportunity to present our results. I will change to my to remove the caps, yeah. Let me try. Can you come here? Let me change the camera, or actually Katrin is perfectly aligned. So first things first, what is ideal, and then what I showed you in the other lecture is how the amplifier can just be put away, and that's it. And if you had the second subject, you can just go and connect to the other subject. To remove the cap is very easy. You just need to take care that you remove first the reference, just so that you don't pull the cables, because pulling the cables is not ideal. And then you just remove the chin strap. You take from the front so that the person doesn't get any electrode into the eyes, and that's basically it. So Catherine has a bit of gel, but not too much, so we can always clean the surface with a paper towel, for example, so that the excessive gel will be removed, so that you look nice again. And that's basically it. Never forget to clean the ears as well. And that's basically it. Subject is happy, we are happy. Okay. We can show also Evgeny as well, so that we can show you also the new magnet. So as you could see, Katrin had a holder with the new version of the caps. We have a magnet holder where you just simply pull out. So you can see how easy it goes in and goes out. You just remove it. In the same way, remove always the reference, remove the chin strap, pull it a bit from the front so that you don't have any cables going into the eyes. And that's basically it. That sounds very neat. Thank you so much for your help. Back to you, Christoph. We can still not hear you. I think we're both having issues on
It's about
the.
now.
Now it's perfect. Now it works.
Mhm.
Okay.
So it's really amazing how clean and stable the EEG recording is. Keep in mind this was nearly impossible a few years ago to do it during table tennis, and nowadays it's not really a problem anymore. We have some questions. So Baide is asking, is FNIRS maybe better for sports applications than EEG?
FNIRS in terms of noise artifacts is definitely less sensitive as EEG. The main thing that the EEG as superior to FNIRS is the time resolution. So for FNIRS, you will have a reaction across longer time scales. So many of the things that we see that are being applied to sports studies, for example, evoked potentials, will not be able to be seen with FNIRS.
Next question is about ethics. You know, there are some people who have too much money and other people who don't have enough. And if there's performance enhancing technology available, some people are able to get it and others not. So how do you see ethical concerns
Ethical concerns in terms of sports?
in
It's a
performance enhancement with
okay.
new
Okay.
technology?
Yeah, that's a very difficult but very important question. I think many institutions are trying to cave in into this question. Also, institutions like the European Union is trying to go into detail about this question. In most of the cases, if you are thinking about enhancement of performance, what we are often seeing is how the EEGs, and especially tools where you have some feedback, can actually help you to get better results in concentrating, or for example in being able to relax, which are very important in these kinds of studies. This is not too much different from other techniques like meditation or something like that, but it's something that we have more biological signal to work with and also more proof to work with. So in terms of helping the subjects, it just helps them finding their own ways and speeding up the process of getting better in terms of relaxation and concentration.
Gerhard is asking, is it possible to measure or simulate movement artifacts beforehand and afterwards you can just subtract them?
It will be very difficult because there will be a lot of different kinds of movements that will be performed, and each direction will provoke different kind of artifacts and also the intensity of the artifacts. What we are using in Oscar is something very similar to what you are talking about. So you can predict that some of the drifts on the EEG are caused by artificial by artifacts from movement because they behave in a certain way. You can predict that and use this prediction for that. But having a completely clean based on pre-done artifacts will be very difficult because the artifacts and the technology and the G-force will be always different on the movements.
But much better is, of course, to remove or to avoid artifacts at all. And this is something that we are always doing to an extreme. So we make systems as lightweight as possible. We put everything on the head, and when your head is moving, then nothing is pulling on the electrodes because it's so lightweight. And it keeps the electrode in position, and this just minimizes artifacts a lot. Or we put amplifiers into the electrodes. This helps also a lot. And if you are getting more weight onto your head and you move your head, then of course it's more pulling on the electrodes. And the worst thing is actually a 3D printed headset, because then every single electrode is moving and you just get artifacts. And in the hackathon, you will see if somebody is using a g.tec EEG or a BCI system, then the teams are not talking about artifact removal or artifact cleaning because the data is just clean and robust, pure. And this is exactly what we want. So with some other systems, you have to throw away more than 50% of the data. And this is exactly what you don't want to do, to throw data away, because recording time is money and a lot of effort. So you want to keep every second. Then Colin is asking, do you need any settings for Oscar, or can you just use it right away?
You should typically have an initial phase of resting state of like not doing anything, and then it's applied right away. So you can start the recording, for example, on a Simulink on the block, and just start the model on the unicorn. You just click on it, and then it works straight away.
So we need about 5 to 10 seconds of clean EEG data, and then you can go ahead. So don't start with artifacts, start with clean data, and then people can move and jump and so on. Arthur is asking, why is table tennis the most used sport for this kind of studies?
It's mostly because it's still a sport that you do indoors, which already is one of the issues of the EEG. How the EEG behaves outdoors can be a bit tricky. It's sports where the people do a lot of movements, but still not far away from the source of the recording. We've seen a lot of these kinds of applications, mostly because it's easy to find people and athletes that want to improve their performance and want to be measured and want to improve their quality of the sports, and also because universities have easiness into getting into these sports and doing it.
Somebody is asking about real tennis. Do you imagine you can also use it doing, the big one?
Yes, absolutely. Actually, it was a plan last year, but it started to rain a lot. So we had to pass the task to indoors because we cannot control the weather yet. But it's definitely possible. You can have a PC or the best would be that you would have a tablet on the back of the subject, but you can also have a PC close to the middle and you're communicating with both people, and then you're doing a kind of hyper scanning with two users into one, which is something that is very neat, and we could prove that it also works very good today.
Remember the video that Frankie showed before with Martin doing the backflip? So in this case, we get about 5G. So there's much more acceleration on the head than what you get doing tennis playing. And many headsets would just fly away. So the Nautilus and the Unicorn, they stay on, and you can still measure EEG data. This is just fascinating. So it's opening in a lot of new applications. And what we showed here with the table tennis is pretty unique. So you will not find many setups where this is actually working without artifacts. And now we are already 2 minutes over time. Thank you very much, Frankie. Please
Welcome,
stay a little
welcome.
bit and answer
Yes.
the questions in the Q&A.